Good evening, afternoon, and evening, everyone. It's my great pleasure to being here and welcome you everyone for today's session. In today's session, we will share our ideas and our work here. So let's not delay any time. So let's begin. So for today's session, I would like to invite Prabha Sharma to present her work here.
Very good morning to everybody. I am Dr. Prabha Sharma, a former guest faculty, Department of Botany, University of Delhi. I thankful to the Society for the Study of Evolution for giving me this opportunity to present in the virtual meeting of Evolution 2026. I wish the meeting to be a grand success, both virtually as well as on site. So I start my topic here as I will be presenting on a thought-based plus experimental work. So the topic of my talk is in urgent need, promotion of plant taxonomy and collection-based research, a stepping stone for novel foundations of technologies supporting plant evolution. So I will be try to convey how the traditional science can be linked to the modern science for the benefit of the society. So let's begin with this talk. I will not give you a wider introduction because in today's era, everybody knows and much of the information is available online. So just briefly, I would like to say that this talk will be emphasizing collection-based research, and there are many different types of the collections. Like mostly we have the dry and wet collections. We also have various libraries which are very renowned and archives which can be used as a collection material. We can have the digital content in the form of audios and videos. Then we also have online resources which is utilized by all over the scientists in n number of ways. So I will be utilizing or I will be emphasizing only on one particular collection based study here, and that is the herbarium. So this is a very prestigious and renowned heritage sites, I would say, and one must preserve these herbarium and the herbarium specimens. Now, what I'm showing you on the screen is I visited recently for my research work one of the two greatest herbaria. One is the Harvard University herbaria, and another is the William and Linda Steer Herbarium at the New York Botanical Garden. Now, I visited this for a specific genera which is called Ephedra. So, I shall be further showing you some of my field study work and how these traditional science can be linked to the field of technology. So, as I said you that I visited this herbarium for a particular genre. For those who are unaware of what is Ephedra it is a gymnospermous genre belonging to the order Ephedralis, and it has got about 50 to 70 species recently, and it is most widely used monograph is of only staff 1889, which is very old dated back and which demands the revisionary work. And that was my thought and my desire in future to work for producing a monograph on this genus. And luckily I got a chance to visit the two herbaria, but I could not spend much amount of time. In future, if I got this opportunity, I would like to collaborate on this. So I request all of those who are listening to me, if they wish to collaborate on this specific topic of monographic study on Ephedra or artificial intelligence based models, I will be very happy to collaborate. but right now I don't have any funds. Now coming back, this genus is very very important because of its ecological nature. It acts as a soil binder, and also it is very very important for the medicinally importance for alkaloids like ephedrine and pseudoephedrine. So this genera is ecologically as well as economically very very important. Here I am showing you the areas of field work which we have covered for our research work. This is particularly from India, and you can see this is a small state which is called Himachal Pradesh. And if you go to Himachal Pradesh, inside you have various districts. So I'm showing you with regard to one particular district, that is Lahaul and Spiti. So we have covered the various localities from Murling to Dakcha and Dakcha, Baralachala up till to the lake. The Western Himalaya is a very wide range, so we have covered a bit of it. But whatever we have covered, we have collected all the populations of this specific genus. And then, as I told you, that doing this research work for revisionary work, as the information is quite old dated back, it was not easy. So here lies the importance of the collection-based research, from where I got much of the information initially to start my work is nothing but the herbaria collection. So I visited various herbaria in India as well as abroad to collect the data, and then I started my work, and then on the basis of that work I also produced I segregated my collection into different populations of various hyperdiverse species, and I also made a herbarium record very carefully, and I documented each and every specimen, and I submitted all those specimens to the Department of Botany, University of Delhi Herbaria. So after doing this much work, then I was able to identify new species. So to publish and to get a new species is again a challenging work, which comes by much efforts and knowledge. Those who have published can understand that this is not an easy work, but that's how I was trying to emphasize that this is a collection-based research, starting from herbaria to the field work and then to publishing the novel Epicra species. So that's how the traditional science is giving birth to the novel species. So we always respect our traditional sciences, and I emphasize the taxonomic-based research to be conducted, and some new grants, if possible, to be given to the plant taxonomists, in particular to the underrated or under-researched genres where the revisionary work is required. Now, this I haven't done any work with respect to technology, but this is my thought, and I would, as I said in the beginning, that I wish to collaborate on these aspects, because nowadays everybody is behind the technologies and it is a good thing. But if we can combine the traditional science with the technology, then I think it could be a plus point. So in the era of the artificial intelligence and modeling, I will emphasize here that if I can combine my traditional science of herbarium studies with the artificial intelligence and modeling, which can predict some of the research predictions in terms of speciation and evolution, or in terms of endangered species with respect to plant conservation, if we can produce some models which can predict or which can show some audios or some videos which can reflect a clear-cut account of the novel species or a clear-cut account of the endangered species, or if we can give knowledge to the upcoming generation in the form of models, or if we can make it online, it will be a good thought. I wish I should produce some models with my research data if I got some opportunity. definitely I would like to work on this aspect. Now, this is regarding my short talk. So, in nutshell, I can say you that novel species, I emphasize the taxonomic research, I emphasize the collection-based research. I also emphasize the novel species because these are the species which can be of huge importance to the industry. I'm not from the industry, but the people who are into the industry, they can utilize this knowledge of medicinal importance of these plants or economical importance of these plants. And various data scientists, they can pull off these results and they can produce drug discovery models also. Not only the drug discovery models, but if somebody is interested to do something related to the indigenous people of the society or the tribal people of the society, where these plants are particularly growing, they can make some good steps for the benefit of the society or benefit of these indigenous people, to make a law or a policy that all the incentive which is going to the industry should also be given to the local tribal people. So this is a very good topic related to the public policy also, if some of the common men are between taking some advantage. Then this is an interdisciplinary work. Various scientists can combine on this work, like herbarium, people working in herbarium, people working in the computational biology, or people working in mathematical sciences, or people working in the field of design and modeling. So this is a kind of interdisciplinary work where many scientists can come and collaborate and produce some good results for the benefit of the society. Not only this, this work, if I produce some good monograph, if I produce some good model with respect to a particular genre which needs a revisionary work, then it will be a plus to the benefit of society. So this is my thought-based study. With this, I acknowledge the travel grants given to me from New York Botanical Garden, William and Linda Steer Herbarium, and Harvard University Herbarium. Botanical Society of America for Developing Country and Professional Member Award, which made this study possible. I also thank the Society for the Study of Evolutionary Biology for virtual presentation and membership. At the end, I thank the project which is granted to me by Indian Institute of Sciences, Bangalore, and Department of Science and Technology, Department of Biotechnology, and University of Delhi, where I work. So with this, I thank you all for giving me this opportunity. Thank you so much.
Thank you, Prabha, for sharing your wonderful work with us. So next, our participant is Lea Ozdere.
Okay, thank you.
Thank you, Prabha.
Thank you.
Hi, everyone. I'll go ahead and share my screen.
Welcome, Liz, and you can start your presentation.
Great, thank you. Can everyone see okay?
Yes.
Perfect, thank you. Hi, everyone. I'm Lea Ozdere from Cal State Monterey Bay. Thanks for being here. Today, I'm going to talk about some of our work in my lab, trying to understand the processes underlying diversification in rockfishes, and particularly the role of depth adaptation and molecular convergence. So rockfishes are a really interesting system because they combine a lot of the features that make adaptive radiations interesting: rapid diversification, ecological divergence, ongoing gene flow, and repeated adaptation across some environmental gradients. So today I'm going to focus mainly on two things that we've been studying. First, building a more complete phylogenetic framework for this genus. And then second, looking at visual adaptation and specifically rhodopsin evolution across depth gradients in the radiation. I'm not going to spend too much time on this general background since this audience knows the lit very well. But a lot of modern speciation research is really centered around understanding how genomic variation, as well as ecological divergence and phenotypic evolution, interact during the process of diversification. And adaptive radiations are especially useful because they let us study these processes over relatively recent evolutionary timescales. So many of these systems that'll be familiar to you, like cichlids, anoles, Darwin's finches, we repeatedly see similar questions emerging. First, how do species form in the face of ongoing gene flow? So this is particularly tricky in marine environments where there are no obvious barriers to gene flow. What's the role of ancestral variation in adaptation, and particularly when it comes to incomplete lineage sorting and deep coalescence? And then finally, does speciation result from selection that generates islands of genomic differentiation? So do ecological radiations involve a relatively small number of genomic regions that repeatedly differentiate across species pairs? So these are some of the kinds of questions that we're ultimately interested in the genus Sebastes, the rockfishes. So rockfishes are a particularly nice system for these questions, include over 110 species, primarily in the North Pacific, with many species living in sympatry, but they occupy very different ecological niches. So they span a huge depth gradient, moving from shallow kelp forests and rocky reefs down to deep continental slope habitats. And importantly, many of them are ecologically similar enough that we can make meaningful comparative genomic comparisons, but divergent enough that we can ask how ecological adaptation contributes to lineage diversification. So in a lot of ways, they function as sort of a marine analog to the African cichlids. And this figure from Roboski and colleagues is one of my favorites because it really highlights how exceptional rockfishes are evolutionarily. So they fall among the fastest diversifying fish radiations, both in terms of speciation rates and phenotypic evolution, in the top 10% of fish clades here. The terminal branch lengths in this figure are proportional to speciation rates. The tip size and colors here represent extant species richness and the rate of body size evolution. Much of the diversification in rockfishes, as we'll see, appears to be tied to ecological opportunity across depth and habitat gradients. So the group really gives us an opportunity to look at how ecological divergence unfolds in this relatively recent and ongoing marine radiation. One of the hypotheses we're interested in is whether that adaptation to different depth environments has contributed directly to rockfish speciation. As I mentioned, they occupy an enormous depth range. So depth is really a multi-dimensional ecological gradient. As you move deeper, not just light decreases, but temperature changes, pressure increases, prey community shifts, habitat structure shifts. So depth potentially creates this very strong axis of divergent selection. Many rockfishes also show ontogenetic habitat shifts, where their habitat use changes through their life history. So juveniles might occupy somewhat shallower habitats before moving deeper as adults. And many species become increasingly specialized ecologically with age. So over evolutionary time, this depth-associated habitat sorting could reduce gene flow and contribute to reproductive isolation. One of the things that complicates our understanding of rockfish speciation is that the phylogeny for Sebastes is not terribly well resolved. There have been a number of studies that have produced phylogenies, but they recover substantially different topologies, especially for those deeper nodes and relationships among the major clades. So here I'm showing examples from Heidenvetter, Kalora et al., and Bescher et al. Some of the clades are recovered consistently, others move around quite a bit depending on the data set and the method. The reasons for this are several-fold. First, diversification appears to have been relatively rapid, so that leaves short internal branches. Second, of course, incomplete lineage sorting is probably widespread, as it is amongst many adaptive radiations. And third, there's growing evidence for integration and hybridization among lineages. And these studies also, I should point out, used very different data types. So from mitochondrial loci to whole genomes to microhaplotypes. And those data sets might capture very different evolutionary timescales and histories. So this matters because having this phylogeny unresolved makes it a lot harder to identify true sister species and reconstruct how traits like that depth adaptation have occurred. So for our research goals, our work is trying to address these questions from a few complementary angles. First, we want a better resolved phylogeny for comparative analyses. We want to test whether unrelated or distantly related deepwater species show evidence of convergent adaptation. And third, we want to identify genomic regions and candidate genes associated with ecological divergence across those depth gradients. So today I'm going to be focusing mainly on phylogenetic work and our rhodopsin analysis. So to start with, to improve that phylogenetic resolution, we have been generating a UCE, ultra-conserved element-based phylogeny using whole genome, low coverage whole genomes from 79 species that were already available on NCBI, along with additional sequencing from 8 more taxa in our lab. So we started with 500 UCE loci, and then we filtered down to 127 in order to maximize our data completeness for each taxon. And we included substantial flanking sequence around each UCE, so about 5 kb on each side, because those conserved cores themselves are, they often contain lower phylogenetic information at those shallow timescales. So this produced about 1.3 megabases of aligned sequence, which we then used to infer both maximum likelihood tree and a Bayesian phylogeny. And here we have our preliminary tree from the UCE data. So we have a lot more to add to this tree, both taxonomically, and we'd like to improve the number of UCEs we use as well. And we also want to include multiple specimens for species where possible. But in our preliminary tree, it helps resolve several outstanding questions in the phylogeny. So here I've shown species that are deep dwelling with a blue dot. We define those as species that have a mean adult depth that's greater than 200 meters. And what you can see here is that deep and shallow species are both distributed across the tree. There are some clustering, but one thing that's interesting about sebastes is that deep water living is thought to be ancestral, while shallow water specialization appears to have evolved repeatedly. So that repeated ecological transition makes the group especially useful for testing convergence. And speaking of which, so one trait system that's especially relevant to depth adaptation is vision. As light penetrates the water, the spectrum changes dramatically, and that creates strong selection on visual systems. So rhodopsin, or RH1, is the rod opsin responsible for dim light vision in vertebrates. And across many, many vertebrate systems, including deep sea fishes, cichlids, bats, and marine mammals, RH1 has shown positive selection associated with low light environments. And importantly, decades of work on both RH1 and other opsins have identified key spectral tuning sites where relatively small amino acid substitutions can shift wavelength sensitivity. So it really provides an interesting candidate gene for studying molecular adaptation to depth. In our analysis, we built on a prior study by Sivasunder and Palumbi that looked at RH1 in 32 rockfish species. And we brought in this to 81 species, spanning shallow, intermediate, and deep habitats, and including Western Pacific and Atlantic species as well. We reconstructed gene trees, tested for positive selection, and used codon-based models in PAML to do this. We also screened available genomes for any evidence of RH1 duplication. So what did we find? There is, of course, strong constraint on this gene, but across 247 amino acid positions, we found 20 to be variable, and almost all those variable sites occurred in transmembrane helices. So that's consistent with patterns that are seen in other teleosts as well. We found no evidence for RH1 duplication in sebastes. So adaptation here appears to occur primarily through relatively small amino acid substitutions within this single conserved copy. And the RH1 gene tree itself shows fairly low resolution overall. That's not surprising for this highly conserved gene, but we can still see this interesting ecological signal. As you see here, the deep water species, which are shown in the darker blue, they tend to cluster more strongly, often in ways that don't match our UCE phylogeny, while the shallow species they often show shared haplotypes across lineages. So that could reflect incomplete lineage sorting, integration, or perhaps balancing selection in shallow environments. And this picture gets even more interesting when we look at the Western Pacific rockfishes. So these are species that are found in Korea and Japan in the whole genome and UCE phylogenies. These are found to be a proper clade. But here we can see that the deep water Western Pacific taxa are clustering with our deep water Eastern Pacific species, whereas the shallow Western Pacific are clustering with some of the shallow Eastern Pacific. So again, this is suggesting to us strong convergence at the level of the RH1 gene, And indeed, we've seen the same amino acid positions that are variable in the deeper species to be consistent across these geographic regions. When we test for positive selection, we see, interestingly, deep water taxa have fewer positively selected sites overall, but much stronger selection coefficients. So the omega values for very deep taxa were really high, around 10 to 16, with very strong statistical support. And the sites identified under selection were all associated with depth-related lineages. So rather than broad divergence across the gene, what we're seeing appears to be relatively targeted selection acting on a small number of functionally important residues. This schematic will help place those selected sites into structural context. So this is a schematic of the RH1 protein. The purple boxes are the transmembrane helices of RH1, and the outlined circles correspond to previously identified spectral tuning sites from other systems like cichlids. The colored dots show variable and positively selected sites in the rockfish dataset. And what's striking is that many of the positively selected sites that we identified occur directly adjacent to predict previously known tuning sites, including those associated with depth adaptation in other fishes. So even though RH1 is highly conserved overall, the substitutions that do occur are concentrated in these regions that are already known to influence spectral sensitivity. So that strongly indicates these substitutions are functionally meaningful rather than random drift. All right, so to wrap up, there's a few main takeaways. First, this revised phylogeny helps stabilize several relationships within sebastes, and it helps us provide a better comparative framework for studying ecological divergence. Second, RH1 evolution in rockfishes seems to reflect a balance between strong structural constraint and repeated adaptive convergence. And third, depth appears to be a highly repeatable axis of molecular adaptation across the radiation. So even within this very conserved gene, relatively small numbers of substitution can produce potentially important functional changes. So going forward, we'd like to expand this work to additional opsins and ideally test spectral tuning directly using protein expression assays. And we're also increasingly interested in connecting these molecular patterns to broader questions about genomic architecture and speciation across depth gradients. So with that, I just want to thank the many collaborators and students, NOAA partners, and lab members who contributed to this work. And a lot of these data sets were only possible because of long-term sampling efforts and collaborations across institutions. So thanks very much, and I'd be happy to take questions.
Thank you, Liz, for your wonderful presentation. If anyone have any questions or anything, you can ask directly. And our next presenter is Lea Ozdere. In the meantime, anyone wants to ask anything, you can ask.
Looks like there's a question in the Q&A from Zachary. Are the limits in UCEs due to particular taxa that enriched poorly?
There are, yes. So both that in some cases, and then also because some of the genome assemblies that we were using were very low coverage assemblies, there were cases in which we just couldn't identify the UCs in those assemblies. Great question. And then I see another one. Have you used something like PhyloAct to look at your system? We have not, but that's a great idea. Thank you. I will definitely look into that.
Thank you for today's session, and we will move to Lea Ozdere. Lea, you can start your session.
Hello, everyone. Thank you for having me. My name is Lea Ozdere, and today I will be talking about how postzygotic barriers can be maintained and even reinforced in early speciation under high gene flow. So postzygotic barriers are inherently underdominant, meaning that they produce less viable offspring and therefore suffer a fitness disadvantage, causing the alleles to be selected against. Classical theory therefore says that they cannot persist or evolve under gene flow. However, we keep finding instances of postzygotic reproductive isolation in sympatric gene-rich environments in the real world. So we look into how these can emerge and even reinforce in early speciation under high gene flow. Before we get to postzygotic barriers, I'll introduce the initial modeling framework that we adapted to apply to our current topic. In a previous study, we developed a mathematical model with a 2 mating bias allele system. We have 2 niches, niche A and niche B, each with 2 alleles X and Y. In each generation, individuals undergo ecological selection and then sexual selection to produce viable offspring. We have 2 variables, F and A. F is the offspring return ratio, which is also the fraction of inter-niche mating offspring that come out as viable parental ecotypes. The lower value of F means more hybrid loss and therefore stronger disruptive ecological selection. And alpha controls mating bias and pre-mating reproductive isolation, with a lower alpha meaning stronger reproductive isolation. We developed a computer application using MATLAB to solve this mathematical model. This interface allows us to change variables and have real-time feedback to visualize the solution as a phase portrait and track our allele frequencies. We can see all of our relevant variables over here, for example, F and alpha that we just mentioned earlier. In the phase portrait, we have AX plotted versus BX, and the x-axis is essentially the frequency of allele X in niche A. Since all of our parameters here are normalized, the frequency of allele Y is 1 minus X. And the y-axis is the same with alleles X and Y for the niche B. The blue dots are the initial trajectory, the starting populations, and each trajectory is one simulation starting from a different initial allele frequency. As we change the parameters, we can see how the phase portrait changes. When the trajectories run into a corner over here, this means that one allele is completely dominating. However, when they converge to a fixed point, seen by this red dot, that represents a stable polymorphism, meaning niche A is mostly allele X and niche B is mostly Y. Whenever we see that red dot move closer to the right lower quadrant of the graph, we are maximizing reproductive isolation, which is what we want to see. Now we adapt that model to postzygotic barriers to demonstrate hybrid incompatibility. Once again, there are 2 ecotypes, A and B, which are analogous to the mating, each with 2 incompatibility loci P and Q, and this is analogous to the mating bias alleles X and Y. Each loci carries 2 alleles in each niche, P1, P2, and Q1, Q2, and both of these alleles have to be present for a fully functioning situation in each niche. However, when there's inter-niche breeding, and we get hybrid offspring that mismatch these allele combinations, we lead to reduced hybrid viability. This is compatible with the classic BDM incompatibility mechanism. So in our new model, we have P represent the pair of alleles P1 and P2, and Q represent the alleles Q1 and Q2, and these replace our mating bias alleles X and Y. The hybrid offspring are incompatible, and we use the variable V instead of alpha now to convey the ratio of offspring that survive, also known as hybrid viability. We can use the same phase portrait application that we showed earlier to find new solutions to our new mathematical model simply by changing the variables. As we can see, now our axes are AP and BP to represent the frequency of those alleles, with Q being 1 minus P. And we also see our hybrid viability variable V here. With this model, we started by seeing how the level of gene flow affects postzygotic reproductive isolation. On the left, we have a high gene flow environment represented by an F value of 0.5, and we see that the trajectories move to the corners of the plot, which means that one incompatibility allele drives the other into extinction. So on the bottom left, this means that allele P is completely eliminated, and up here, this means that allele Q is completely eliminated. The phase portrait on the right shows a 0 gene flow situation with the value of F set to 0. And here, each niche evolves independently. Since there is no gene flow, there cannot be any reproductive isolation. Therefore, neither of these extreme cases produces stable postzygotic reproductive isolation. However, when we set gene flow to an intermediate value, like F equals 0.1, we see that the trajectories converge to a single fixed point in the right lower quadrant. This fixed point means that niche A is mostly allele P and niche B is mostly allele Q, and gene flow cannot homogenize them. This little dot represents that, and we only see it in the right lower quadrant, means that this only happens when there's already previous divergence. A scenario like this can be among secondary contact when geographically isolated populations have already accumulated incompatibility alleles and contact each other again. When we look at the left lower quadrant, we see that these trajectories still converge to 0, meaning that the mutation cannot invade from the origin because it is underdominant. But with reduced gene flow and disruptive ecological selection, we see the development of postzygotic reproductive isolation. Now that we've established what level of gene flow causes the emergence of a reproductive isolation, we look into what conditions strengthen or weaken it. And the first of these is investigating disruptive ecological selection. On the left, we have an F value of 0.125, and we see that there is no fixed point on this graph. However, when we decrease F even just slightly to 0.1, we are therefore strengthening the disruptive ecological selection, and we see the development of a fixed point in the right lower quadrant. If we were to further decrease the value of F, the stable point would move further and further into the corner, signifying increasing postzygotic reproductive isolation. Greater incompatibility strength, also known as lower hybrid viability, acts very similarly. When V, the variable that we use, is high, this means that hybrids are nearly as fit as their parents. This means there's little selection against mismatched combinations and no fixed point forms. However, when V drops, meaning that now hybrids are significantly less viable, we see that selection pressure grows and the system locks into a stable lower right fixed point. Next, we look at pre-mating mating bias reproductive isolation. With no mating bias, seen on the left, the fixed point sits in the interior of the face portrait, representing partial reproductive isolation. However, when we add a mating bias barrier on the right, we see that this fixed point drives deeper into the right lower quadrant, signifying increasing postzygotic reproductive isolation. Another pre-mating barrier is habitat preference reproductive isolation, and the same logic as the previous slide applies to this mechanism. Like mating bias, when we add habitat preference reproductive isolation, we see the fixed point drive deeper into the corner and therefore strengthen. Niche size is another factor, and when we see that we have roughly equal niche sizes, we produce the maximum amount of offspring and therefore have a fixed point. But as these niche sizes become more asymmetric, we lose the stable post-zygotic reproductive isolation. And lastly, for our strengtheners, we find that when we have an initial postzygotic barrier introduced, it facilitates the coupling of the second one because the invasion of the first barrier reduces gene flow, which creates an environment that makes it easier for following postzygotic barriers to emerge. So the foundation of the mechanisms behind everything we just saw is selection migration bias, and this diagram provides an alternate view without using phase portraits, but applying the same concepts. Within each niche, there is a predominant allele, and the selection removes the foreign minority allele. However, migration of alleles continuously reintroduces the foreign allele from the opposing niche. For example, in niche A is predominantly P, and it's working to constantly eliminate Q. However, migration of alleles from niche B into niche A constantly reintroduces Q to A. The ratio of these forces bases the frequency of the alleles in each niche. And when there is a balance between these forces, that's when we see the stable polymorphism. If anything were to disrupt this balance, that's when we'd see a shift. And the 6 points that we previously just walked through, this is how they work. They either lower effective migration, also known as gene flow, like pre-mating barriers and disruptive ecological selection do, or they raise the effective selection, which lowering higher viability does. And all of these strengthen post-zygotic reproductive isolation. These are the 3 isolating mechanisms we studied, and we discovered that there is reciprocal reinforcement and positive feedback between each of them. For instance, we already know the classical reinforcement model of postzygotic incompatibility driving the evolution of pre-mating isolation, but now we've demonstrated that this also works in the reverse direction, with pre-mating barriers strengthening postzygotic reproductive isolation, forming a bidirectional feedback loop, which we confirmed by our simulation results that we previously just showed. The same relationships exist between each pair of these 3 isolating mechanisms. And now we'll look at what happens when we move from a single locus barriers to polygenic reality of these processes. Even though we've treated each barrier as governed by a single locus thus far, in reality, these involve multiple loci, and the effect of the number of loci depends on the mechanism. In general, the presence of more viable hybrids will always weaken the selection pressure. In the case of disruptive ecological selection, more loci produces more maladaptive hybrid offspring, and this creates stronger ecological selection. Similarly, with hybrid incompatibility, more loci creates more mismatched hybrid combinations, also increasing inviable hybrids, and then creating stronger postzygotic isolation. But in contrast, mating bias, the more loci that you have, the more viable hybrids you produce, which therefore weakens its selection. And the effect of loci number is how chromosomal inversions have their effect on these systems. Chromosomal inversion locks multiple loci into a single unit, and by doing so, they're effectively decreasing the number of loci. Therefore, a chromosomal inversion can either promote or hinder development of reproductive isolation, depending on how the number of loci affects the selection strength of each mechanism. We see that with a chromosomal inversion that captures disruptive ecological selection or hybrid incompatibility, the effect of the inversion will weaken reproductive isolation because it is decreasing the number of loci, and we know that more loci creates stronger selection for these mechanisms. In contrast, as discussed with mating bias, a chromosomal inversion will lead to stronger reproductive isolation because it is reducing the number of loci. We also found that when a single chromosomal inversion captures alleles from multiple mechanisms at once, reproductive isolation is always strengthened. We can see that when this chromosomal inversion fully invades the population, our fixed point drives deeper into the right lower quadrant, signifying increased postzygotic reproductive isolation. And the genotype distribution on the right confirms this with the allele frequencies in the niches. So if we wanted to maximize reproductive isolation, we would capture as few ecological and incompatibility loci as possible and as many pre-mating bias loci as possible. So in conclusion, we've shown that low gene flow plus disruptive ecological selection allows for the emergence of stable polymorphisms, and we talked about the 6 strengtheners that further strengthen this. Reinforcement is bidirectional with pre and post mating barriers, developing a reciprocal positive feedback loop. Locus number strengthens ecological and postzygotic selections, while it weakens pre-mating selection, and chromosomal inversions can harness this to have that effect. And single mechanism CIs can hurt or help depending on which allele, but whenever a chromosomal inversion couples alleles from different mechanisms, the result is that the reproductive isolation is always strengthened. If you are interested in learning more or reading deeper into this topic, here's the QR code of the preprint for the paper. And I want to thank my lab members and collaborators that contributed to this project. And thank you for having me.
Thank you, Lea, for your wonderful presentation. And if anyone have questions or anything, you can ask using, yes, we already got one. Your question is there any reason to believe that these models would differ for a strong prezygotic reproductive isolation, given that disruptive ecological selection can lead to behavioral and habitat isolation?
So sorry, could you repeat the first part of the question? Is there reason to believe that stronger reproductive?
A strong prezygotic reproductive isolation, given that disruptive ecological selection can lead to behavioral and habitat isolation.
Yes, certainly. So that goes into the feedback loop that we mentioned in reinforcement. So strong prezygotic isolation does definitely further and strengthen postzygotic reproductive isolation.
Anyone have more questions? Okay.